Today we are going to explore the surprising story of a medical recipe, over 1,000 years old, hidden within the yellowed pages of a medieval manuscript that modern medicine had practically ignored for centuries, until an unlikely group of scientists decided to test it in a laboratory and made a discovery that shook the foundations of contemporary microbiology. Before continuing with the video, remember to subscribe to the channel for more documentary narratives like this. It was the year 2011, and the corridors of the University of Nottingham in the heart of England were inhabited, as always, by hurried voices, stacks of books, and the familiar aroma of old coffee mixed with the characteristic scent of study rooms. Nothing in that common setting heralded what was about to happen. It was there that a medievalist with a measured voice and attentive eyes, specialized in Anglo-Saxon texts, began to ask a question that seemed improbable. What if the medical recipes she studied, written in an English so old that it sounded almost like a foreign language, contained something real? Something that modern science had never stopped to verify? Her name was Christina Lee. A researcher dedicated to the languages and cultures of the early Middle Ages, she spent her days among manuscripts from the period stretching, approximately, from the 5th to the 10th century AD. To most people, this was a distant and opaque time, associated with knights, superstitions, and intellectual obscurity. But to Dr. Lee, it was a world of surprising richness, filled with human details, practical knowledge, and wisdom accumulated generation after generation that modernity had decided to ignore, almost without bothering to question that decision. In her research, she repeatedly came across medieval medical texts, manuscripts containing hundreds of recipes and remedies for the most varied conditions: fever, headache, battle wounds, digestive problems, inflammations, and infections. These texts mixed botanical ingredients with precise preparation instructions, timing indications, and at times ritual elements or prayers. For this reason, modern science had treated these sources as historical curiosities, relevant to anthropologists and cultural historians, but certainly of no value to practical medicine. Dr. Lee began to suspect that this conclusion, so automatically accepted, had been adopted without a real test, without anyone having verified with rigor whether those recipes actually worked. It was from this suspicion that she crossed a university hallway and knocked on the door of a microbiology laboratory. And it was there that she met Dr. Freya Harrison. Harrison was a professor at the School of Life Sciences at the University of Nottingham and had dedicated years to the study of pathogenic bacteria, those capable of causing serious diseases in human beings. Her area of interest included, in particular, infections caused by methicillin-resistant Staphylococcus aureus, better known by the acronym MRSA. This pathogen had become, over the last few decades, one of the greatest challenges of modern hospital medicine. Treating it was increasingly difficult. Finding new approaches to combat it was a growing urgency. When Dr. Lee presented the idea of testing medieval recipes in the laboratory, Harrison could have smiled politely and ended the conversation. But she did not. On the contrary, she saw in the proposal something that few people would have recognized there: a genuine opportunity for scientific investigation in a completely unexplored territory. Together, the two researchers launched what would become known as the Ancient Biotics Project. The initiative was ambitious and methodologically challenging to select medieval recipes with some potential therapeutic relevance, recreate them with the greatest possible historical fidelity, and subject them to rigorous laboratory tests with reproducible conditions and appropriate controls. It was an endeavor that required simultaneously the rigor of experimental science and the depth of historical and linguistic knowledge. It was, in short, a project that did not fit perfectly into any existing discipline precisely because it crossed all boundaries. The manuscript that captured the researchers' attention from the beginning was a volume now preserved at the British Library in London, one of the most important and well-preserved institutions of documentary memory in the world. The manuscript had arrived there through a journey of centuries, passing through the anonymous hands of copyists, surviving wars, fires, and indifference. It was known as Bald's Leech Book, in reference to the physician who, according to the text itself, had commissioned its compilation. The English word leech, in modern English, means a blood-sucking worm, but in Old English it simply designated a physician or healer, someone who practiced the art of healing. Bald's Leech Book was, in essence, a 9th-century medical compendium, a manual of health practices written during the reign of King Alfred the Great of Wessex, around the year 900 AD. The manuscript survived the centuries in a state of admirable preservation. Its parchment pages, covered in an angular and regular handwriting, contained hundreds of recipes for a wide variety of conditions. The language was Old English, a tongue so different from modern English that to the current reader it seems like a completely foreign language. Deciphering its texts with precision required years of specialized study and constant care regarding the ambiguities inherent in translating historical sources. A misinterpretation could completely compromise the reliability of a recipe and render the results of any test based on it irrelevant. Dr. Lee led this work with the meticulousness that the profession of medievalist demands, and that few outside the field can fully grasp. Among the hundreds of recipes in Bald's Leech Book, the researchers selected one that described a remedy for a condition called, in Old English, sty on eye. What we call in Portuguese a tersol, sty, a common bacterial infection that affects the eyelid glands, causing redness, swelling, and pain. A sty today is treated with topical antibiotics or warm compresses and rarely causes serious complications. But in the Middle Ages, any infection, however apparently simple, could become serious without proper treatment, and the medicine available to combat it was exactly that which was preserved in manuscripts such as Bald's. The selected recipe was, at first glance, simple. It combined 4 main ingredients: garlic, onion or leek wine, and oxgall. These ingredients were to be mixed in specific proportions, placed in a container made of brass or bronze, and left to ferment for 9 nights before being strained and applied. Anyone in the Middle Ages living in a rural environment in England, or even in many other parts of Europe, could gather these ingredients without great difficulty. They were everyday components, present in the kitchens, vegetable gardens, and livestock pens of the time. There was nothing exotic or rare about the formula. It was, in every sense, a folk recipe, intended for common use. To the modern scientific mindset, this combination would sound strange. Wine and garlic, oxgall stored in a bronze vessel? Initial skepticism was understandable and perfectly reasonable, but the researchers deliberately decided to suspend such judgment and treat the recipe as they would any laboratory protocol, following every detail of the instructions with the utmost fidelity possible, without adding or omitting, without modernizing or simplifying. If the results were negative, they would be negative. But at least they would be results based on a real test, and not on the automatic assumption that medieval texts had nothing relevant to offer. Gathering the ingredients was the first practical challenge. The wine needed to be of a variety compatible with what would have been available in 9th century England, a wine produced from European harvest grapes without modern industrial processes. The oxgall, an ingredient that certainly does not appear on typical 21st century shopping lists, had to be obtained from specialized butchers who still handled parts of animals less common in conventional trade. The garlic and onion were easily found, but the issue was ensuring proportions that were as faithful as possible to the manuscript's indications, taking into account the inevitable inaccuracies of a medieval text translation. The bronze or brass vessel deserved special attention from the researchers. It might have seemed like an irrelevant detail, an aesthetic preference, or a question of the availability of materials at the time, but there was reason to suspect that this detail was significant. Copper and its metal alloys, such as brass and bronze, are known for their antimicrobial properties, something that modern science has documented extensively throughout the 20th century. Copper surfaces are used in some contemporary hospital environments precisely for their ability to inhibit bacterial growth. By specifying a bronze vessel, the medieval healer who developed the recipe might have incorporated, even without knowing it, an element that actively contributed to the mixture's efficacy. With all the ingredients gathered, the researchers began the preparation process exactly as described in the manuscript. Each step was documented with precision, recorded and recorded again, to ensure the reproducibility of the results. The mixture was left to rest during the stipulated period of 9 nights, under controlled temperature and environmental conditions. At the end of this period, the resulting liquid was carefully filtered and properly stored for laboratory testing. It was a procedure that any medieval healer would have recognized as routine. In the context of a modern microbiology laboratory, it was something that no one had done before. The test was conducted against MRSA biofilms, methicillin-resistant Staphylococcus aureus. Biofilms are bacterial structures of remarkable complexity. Unlike bacteria that float freely in a solution, bacteria organized into biofilms form highly structured communities, protected by a matrix of polymeric substances that they produce themselves. This matrix acts as a shield, preventing external agents, including antibiotics, from reaching the bacterial cells in concentrations sufficient to kill them. Testing any substance against MRSA biofilms is, therefore, one of the most rigorous challenges that can be imposed on an antimicrobial agent. It was exactly this level of demand that the researchers wanted to impose on Bold's recipe. The results arrived with a clarity that surprised even the researchers themselves. The medieval mixture, prepared with ingredients that any healer from the 9th century could have gathered in an afternoon, destroyed approximately 90% of the bacterial cells in the tested Marseille biofilms. 90%. In biofilms, notoriously resistant structures that defy the vast majority of conventional treatments. The performance was comparable, and in certain parameters superior, to that of antibiotics used clinically to treat Marseille infections. Bold's leech book had preserved, for over 11 centuries, a formula that modern science was barely beginning to understand. The question that emerged immediately was, naturally, the most fundamental of all. How? In what way could a mixture of such common ingredients, prepared by such rustic means, be so devastatingly effective against one of the most resistant and dangerous bacteria in contemporary medicine? The answer was not simple, and did not come all at once. It was carefully built, piece by piece, as the researchers and their collaborators delved into the biochemistry of the mixture with the tools that modern science offered. Garlic was the most obvious starting point. The antimicrobial properties of garlic had been documented by science throughout the 20th century, although they were often treated as a curiosity without serious therapeutic application. The most studied active compound is allicin, a molecule that forms when a garlic cell is crushed or cut. This happens because allicin, present in the cells of whole garlic, comes into contact with the enzyme alliinase, which converts allicin into allicin almost instantaneously the moment the cellular structure is ruptured. Allicin is a highly reactive molecule, capable of interfering with the enzymatic mechanisms of bacteria, inhibiting essential enzymes for their metabolism and compromising their ability to survive and reproduce. The problem, from a pharmacological point of view, is that allicin is unstable. It degrades relatively quickly under normal conditions, which had made it difficult to use as a therapeutic agent in conventional formulations. Onion and leek, mentioned in the recipe as alternatives, belong to the same botanical genus as garlic, the genus Allium. They share similar sulfur compounds, albeit in slightly different concentrations and forms. The presence of any of these ingredients in the recipe reinforced the total dose of antimicrobial compounds in the mixture, and more importantly, created possibilities for chemical interaction between different molecules of the allium family that could have synergistic effects not present in any of the ingredients in isolation. Wine played multiple roles in the mixture that went far beyond its alcohol content, although the alcohol itself already had recognized antimicrobial action. Wine is rich in polyphenolic compounds, especially tannins, whose bactericidal properties are documented in scientific literature. But the most critical role of wine in the medieval recipe was possibly that of a solvent and stabilizer. The acidic environment created by the wine, with its relatively low pH, created chemical conditions that slowed the degradation of compounds such as allicin and other sulfur derivatives of garlic and onion, prolonging their active life and allowing them to remain biologically effective during the 9-night fermentation period. The wine was not just a carrier liquid, it was an active component of the chemical synthesis process that occurred during the preparation of the mixture. Ox bile was arguably the most unusual ingredient and also the most revealing. Bile is a substance produced by the liver and stored in the gallbladder, whose primary function in the organism is to aid in the digestion of fats through so-called bile salts. These bile salts are amphipathic molecules, meaning they possess both a water-soluble part and a fat-soluble part. This amphipathic property is exactly what defines a substance's surfactant capacity, the ability to act at the interfaces between aqueous and lipid phases, reducing surface tension and, under certain conditions, disrupting biological membranes. In the context of the medieval recipe, the bile acted as an agent that destabilized the plasma membrane of the bacteria, making them far more vulnerable to the antimicrobial compounds present in the other ingredients. Even more crucially, the bile interfered with the biofilm matrix. The protective layer that MRSA constructs to defend itself from external aggression was partially undone by the surfactant action of the bile, exposing the bacterial cells sheltering within to the devastating action of the compounds from the garlic and onion. It was like removing the shield before striking the blow. The 9-night resting period, which could be interpreted as an arbitrary imposition, perhaps religious or ritualistic, revealed itself to have an underlying chemical logic of considerable sophistication. During this time interval, the compounds from the different ingredients interacted with one another in a slow and progressive process of chemical transformation. Unstable molecules were stabilized by the acidic environment of the wine. Precursor compounds were converted into derivatives with even more potent antimicrobial properties. Allicin and its sulfurous derivatives met the bile salts in an environment that maximized their respective activities. The wait was not superstition. It was inadvertently an essential stage of chemical synthesis and maturation, comparable in principle to the incubation time that modern pharmaceutical formulations sometimes require to reach their ideal composition. And the bronze vessel? Analyses performed by the researchers confirmed that copper ions released by the metal during the preparation period. Throughout the 9 days, the mixture remained in contact with the container, also contributed to the antimicrobial activity of the final product. Copper is toxic to bacteria in relatively low concentrations, interfering with their fundamental metabolic processes. Its presence, even in small amounts, added another layer of biocidal action to a mixture that was already formidable due to its other components. What the researchers of the Ancient Biotics Project identified at the end of this analysis was not a simple folk remedy that had hit the mark by fortunate coincidence. It was a formula that combined multiple antimicrobial agents with distinct and complementary mechanisms of action, creating a synergistic effect whose final result was substantially more powerful than any of the ingredients could produce in isolation. This synergy, in fact, is precisely what modern pharmacology seeks when developing combination therapies for the treatment of resistant infections. The fundamental difference is that, in the case of the Leechbook de Bold, this synergy had been discovered empirically, probably through centuries of practical observation and trial and error, long before any scientific, theoretical, or methodological framework existed to explain it, or even to recognize it as such. The release of the results from the Ancient Biotics Project generated a reaction that quickly transcended specialized academic circles. The work was presented publicly in 2015 in the context of the American Society for Microbiology conference and was received with a mixture of admiration, surprise and, among the more cautious, a methodological skepticism that the researchers considered absolutely legitimate and even welcome. Articles about the discovery appeared in scientific publications and the general press, sparking reactions ranging from curiosity to astonishment. Microbiologists working with bacterial resistance saw a new and unexpected perspective in the work. Pharmacologists wondered whether other traditional remedies, discarded decades ago as mere superstition, might hold as yet unexplored bioactive compounds. And medievalists saw, with a satisfaction rarely experienced in their profession, their field of study gain space in the pages of scientific journals dedicated to biology and medicine. To understand why the discovery resonated with such intensity, it is necessary to understand the context in which it occurred. At the beginning of the 21st century, the world of medicine was facing a crisis that was growing in silence, but with increasingly visible and devastating consequences. Bacteria, those invisible organisms that inhabit every surface of the planet, had been in a permanent evolutionary race against the antibiotics that humanity had developed throughout the previous century. And in that race, the bacteria were gaining ground. The 20th century had been, in many ways, the century of antibiotics. The discovery of penicillin by Alexander Fleming in 1928 in a laboratory at St. Mary's Hospital in London was one of the most transformative moments in the history of medicine. That fungus contaminating a bacterial culture plate and dissolving the bacteria around it represented something humanity had never seen before. A natural substance produced by an organism capable of specifically killing bacteria without destroying the host organism in the process. The development of penicillin as a medication, accelerated by the urgency of the Second World War, opened the doors to an era of unprecedented medical achievements. Infections that were once death sentences became treatable with a few days of medication. Pneumonia, tuberculosis, syphilis, gangrene, rheumatic fever, all these conditions that had killed tens of millions throughout human history, began to be fought with relative efficacy through compounds derived from fungi and subsequently chemically synthesized in laboratories. Over the following decades, dozens of distinct classes of antibiotics were developed, each with its specific mechanism of action against different types of bacteria. This pharmacological abundance generated a confidence that, in retrospect, proved to be excessive. The massive use of antibiotics, both in human medicine and in livestock farming, where they were administered to healthy animals as growth promoters, created the perfect conditions for what the theory of evolution had predicted from the start. Bacteria reproduce in very rapid cycles. A single bacterial cell can divide every 20 minutes under favorable conditions, which means that a population can grow from a few individuals to billions in a matter of hours. Each division carries the possibility of a random genetic mutation. The vast majority of these mutations are neutral or harmful to the bacterium. But occasionally, a mutation confers a specific advantage to the bacterium: a reduced ability to absorb the antibiotic, an enzyme that destroys the antibiotic before it can act, or an altered version of the target protein that the antibiotic seeks to bind to, but which no longer recognizes it. When this occurs in an environment saturated with antibiotics, the resistant bacterium survives while its sisters are eliminated. It reproduces and passes its resistance on to subsequent generations. Within a few reproductive cycles, resistance can become dominant in an entire bacterial population. MRSA is one of the most emblematic and terrifying examples of this process. Staphylococcus aureus is a bacterium that normally and harmlessly inhabits the skin and mucous membranes of most human beings. But when it enters the body through wounds, surgeries, or invasive medical procedures, it can trigger severe infections, including pneumonia, endocarditis, sepsis, and deep soft tissue and bone infections. The methicillin-resistant version, MRSA, emerged in British hospitals as early as the 1960s, just a few years after the introduction of methicillin as an alternative to the Staphylococcus strains that had developed resistance to penicillin. The speed with which resistance emerged was a clear warning that the evolutionary race was far from over. In the following decades, MRSA spread through hospitals and communities around the world, and some of its strains developed additional resistance to multiple other classes of antibiotics, making treatment progressively more difficult and, in extreme cases, practically impossible with the available pharmacological options. The statistics associated with MRSA throughout the early 21st century were, and continue to be, alarming. The infection caused tens of thousands of deaths per year in developed countries. In countries with less structured health systems, where early diagnosis and proper treatment were even harder to guarantee, the numbers were likely higher and less precisely documented. The World Health Organization classifies antimicrobial resistance as one of the top 10 threats to global public health, and MRSA, with its combination of virulence and resistance, holds a prominent place on that list of threats. It was in this context of urgency and desperate search for new approaches that the discovery of the Ancient Biotics Project gained its true weight. It was important to make clear, a matter of proposing that modern patients be treated with medieval potions, mixed in bronze containers by healers dressed in Middle Ages attire. The objective was much more precise and much more ambitious from a scientific point of view: to identify which specific compounds present in Bald's recipe were responsible for the observed antimicrobial activity, to understand their mechanisms of action, and from there to pave the way for the development of pharmaceutical formulations that could be subjected to modern safety and efficacy evaluation processes. This path, known in technical vocabulary as bioprospecting, or the search for biologically active compounds in natural sources, was not an absolute novelty in the history of pharmacology. Many of the most important drugs in modern medicine have their roots in substances used in folk medical traditions long before science identified and isolated them. Aspirin, one of the most consumed medications in the world, is derived from salicylic acid, a compound present in willow bark and used for the relief of pain and fevers in folk preparations for millennia, dating back to ancient Egypt and classical Greece. Morphine, the most powerful opioid analgesic in clinical medicine, is derived from the opium poppy, a plant cultivated for medicinal purposes for at least 5,000 years. What the Ancient Biotics Project suggested was to apply this same logic to a universe of knowledge even older and far less explored: medieval European medical texts. There were hundreds, probably thousands, of manuscripts similar to the Leech Book de Bald scattered throughout libraries in Europe and beyond, each containing recipes accumulated over generations of empirical practice. The vast majority of these texts had never been analyzed from a biochemical or pharmacological perspective. They were archives of knowledge that modernity had simply left behind, convinced that nothing concretely useful could be extracted from sources so ancient and so distant from the contemporary scientific paradigm. The question the project posed, with all its unsettling simplicity, was, what if we were wrong? What if medieval healers, over centuries and centuries of trial and error, by meticulously observing and recording what worked and what did not work in their patients, had accumulated genuine empirical knowledge that modern science had ignored, for not knowing where to look or for not having bothered to look? The answer, in the case of the Leech Book de Bald, was right there, in the laboratory results, undeniable and documented. And this recognition carried a significant dose of intellectual humility that science, to its credit, had to accept. To properly place this discovery in its broader historical context, one must visit, however briefly, 9th century England. It was a place and time radically different from the world we inhabit. Anglo-Saxon England was under constant threat from Viking invasions, which swept the country's coasts and rivers in successive waves throughout the 8th and 9th centuries, plundering monasteries, destroying communities, and forcing entire populations to relocate or reorganize in ways that profoundly transformed the social and cultural fabric of the country. In this environment of instability and violence, the continuity of medical knowledge was a permanent challenge. The main repository of this knowledge in that period was the monasteries. Anglo-Saxon monastic communities functioned as centers of education, healthcare, and intellectual preservation in a context where secular learning was fragmentary and unstable. Monks maintained medicinal herb gardens, cared for the sick and the war-wounded, and preserved manuscripts that contained not only religious texts, but also medical works translated from Latin and Greek, originating from the Greco-Roman medical tradition that had survived the collapse of the Western Roman Empire, thanks, in large part, to Islamic transmission and the preservation work of Christian monks in Europe. Bald, the physician whose name gives the Leech Book its title, was likely a doctor associated with one of these monastic establishments. It is possible he was a layman, not a religious, but living and working in association with a monastic community that provided him with the resources to study and compile his knowledge. What we know about him is exclusively what the text itself reveals: that he commissioned the compilation of the Leech Book, probably as a reference for his own medical practice, and that the manuscript that has survived to this day represents, at least in part, the record of a medical tradition that he had accumulated or systematized. Bald's Leech Book was not a totally original creation. Like most medieval European medical texts, it was a composite incorporating elements from diverse sources. Some of its recipes have recognizable roots in the Greco-Roman medical tradition, especially in the writings of Galen of Pergamon, the 2nd century Greek physician who systematized classical medical knowledge into an encyclopedic work that would dominate European medicine for over a millennium, and in the writings of Dioscorides, whose treatise on herbal pharmacology was the most consulted reference in medieval medicine. Other recipes in the Leech Book appear to be creations more directly rooted in the British environment, combining local ingredients and practices with elements imported from the classical tradition. The recipe that the Ancient Biotics Project selected for testing seems to belong to this second category. Its ingredients, garlic, onion or leek, wine, and ox bile, were all components locally available in 9th century England. The method of preparation, involving the use of a metal vessel and a resting period of days, was consistent with practices appearing in other medical texts of the time. It was, in short, a British recipe, likely developed or refined within the local medical tradition and recorded by a physician who had experimented with it or observed it in action. The success of the Ancient Biotics Project paved the way for an investigation that went beyond the initial results. In the following stages of the research, the researchers and their collaborators delved into the analysis of the mechanisms by which the medieval mixture acted upon MRSA. Using advanced techniques of fluorescence microscopy and mass spectrometry, they observed with a precision never before possible how the different components of the mixture interacted with the bacterial cells and with the structure of the biofilm. One of the most intriguing discoveries of this phase of the investigation involved the phenomenon known as quorum sensing. Quorum sensing is a bacterial communication system that remained unknown to science until the late 20th century. Bacteria, far from being purely solitary and independent organisms, communicate with each other through chemical signals, molecules that each cell produces and releases into the environment, and which other cells of the same species can detect. As the bacterial population grows and the concentration of these chemical signals increases, a certain threshold is reached, and this triggers coordinated responses across the entire population. This collective coordination is quorum sensing, and it regulates behaviors essential to the survival and pathogenicity of bacteria, including the formation of biofilms. It is precisely these quorum-sensing signals that trigger the synthesis of the protective polymers that form the matrix of biofilms, the shield that makes MRSA so difficult to combat. Analyses carried out by the Ancient Biotics Research Group suggested that the medieval mixture could interfere with these bacterial communication signals. If confirmed in subsequent research, this property would make the medieval remedy relevant not only as an agent that kills bacteria directly, but also as a quorum-sensing inhibitor, a category of therapeutic agents that modern pharmacology was investigating with growing interest precisely because it offered a way to combat bacteria without simply trying to destroy them, which reduces the selective pressure that drives the development of resistance. A bacterium incapable of coordinating biofilm formation is a bacterium much more vulnerable to the host organism's natural immune defenses and to the conventional antibiotics it had learned to resist. This perspective transformed the way researchers viewed the possible applications of Bald's recipe. Instead of trying to replace conventional antibiotics with the medieval mixture, an idea that would have numerous practical and regulatory limitations, the most promising approach would be to use it as an adjuvant, a substance that, applied in conjunction with existing antibiotics, would make resistant bacteria vulnerable to conventional treatment once again. It was a possibility that, if validated by proper clinical trials, could have enormous implications for the treatment of MRSA infections and potentially other resistant pathogens. The question of safety, as with any potential new therapeutic, would need to be answered with rigor before any thought of human application. The individual ingredients of the recipe, garlic, onion, wine, and animal bile, are substances that have been consumed by humanity for millennia and are present in practically all culinary and medical traditions around the world. But a specific mixture of these ingredients in specific concentrations and after a specific preparation process could have effects on human tissues that would need to be carefully evaluated. Preclinical toxicity studies would be mandatory steps, as would trials in animal models and eventually in human volunteers. before any formulation derived from the medieval recipe could be considered for therapeutic use. The discovery also shed light on the critical importance of preserving historical manuscripts. The Leech Book of Bald exists today as a physical object because it was carefully conserved over centuries, first in the monastic libraries of medieval England and later in the British Library. But how many other manuscripts in other languages, from other cultures and other eras, had been lost forever, taking with them the knowledge they contained? Wars, fires, floods, religious and political persecutions, indifference and neglect over the centuries had destroyed immense quantities of the written record of human experience. What survived was only a fraction of what had existed. The Ancient Biotics Project, by revealing that a medieval European text contained a recipe with demonstrable efficacy against one of the most problematic pathogens in contemporary medicine, placed in a broader perspective a question that modern science had, to a large extent, avoided formulating clearly. How much knowledge had been simply ignored or discarded due to epistemic prejudice? Modern science had developed, throughout its trajectory since the 17th century, an increasingly rigorous criterion for distinguishing valid knowledge from invalid knowledge. This rigor was an immensely valuable advance, responsible for discoveries and applications that radically transformed the conditions of human life. But this same rigor, when applied in an excessively narrow way, had created boundaries that excluded valuable knowledge simply because it had not been produced within the accepted methodological paradigm. Ayurveda medicine of India, which has over 3,000 years of documented history, had been systematically marginalized by Western medicine since the colonial period. Chinese herbal medicine, with its vast repertoire of botanical preparations developed over millennia, was treated with condescension or skepticism by conventional pharmacology. Indigenous medical traditions of the Americas, Africa, and Oceania had been, in many cases, actively suppressed by the European colonial process. In all these cases, a part of the knowledge had been lost or remained latent, without the scientific investigation that could reveal its eventual value. The Ancient Biotics Project did not argue that all this knowledge was equally valid or equally effective. It did not propose an uncritical return to pre-scientific traditions, nor the replacement of evidence-based medicine with untested approaches. It argued, in a much more precise and much more productive way, that empirical knowledge accumulated over centuries deserved to be subjected to the same scientific scrutiny applied to any other potential source of biologically active compounds, that the origin of an idea in a medieval text was not in itself a reason to discard it without testing, and that collaboration between disciplines, between medieval studies and microbiology, between the humanities and the natural sciences, could produce genuine discoveries that neither, working in isolation, would be capable of making. The continuation of the project's research throughout the years following the publication of the initial results expanded the scope of the investigation in multiple directions. New recipes were extracted from Bald's Leech Book and other medieval manuscripts for analysis. The network of collaborators grew, incorporating specialists in organic chemistry, pharmacology, bioinformatics, and clinical medicine. Samples of the mixtures were subjected to detailed spectrometric analyses, allowing for the identification with greater precision of which specific compounds were present and in what quantities, and which were most likely responsible for the observed antimicrobial activity. There is something deeply touching when one looks at the complete history of Bald's Leech Book, in this trajectory of over 1,000 years. A 9th-century physician, whose name has barely survived the passage of time, recorded on parchment the instructions for a recipe that his experience, or the accumulated experience of his predecessors, had identified as effective for treating infections. He did not know what a bacterium was. He had no idea what antimicrobial resistance, quorum sensing, or surfactant action were. He simply knew through observation and practice that that combination of ingredients prepared in that specific way helped his patients. The manuscript passed through the hands of anonymous copyists, was transported from monastery to monastery, survived destruction, traversed time in a way that most objects created by human beings cannot, and arrived intact on the shelves of the British Library. There it waited for centuries, for someone to read it with the right eyes, for someone to ask themselves not only what the text meant, but what it could do. And when that someone finally appeared, in the form of a curious medievalist and a microbiologist willing to listen, the knowledge that the manuscript contained proved to be exactly as valuable as the practical observation of generations had indicated. This is, in essence, a story about the different ways in which human beings accumulate and transmit knowledge. Modern science is, without a doubt, the most powerful method we have ever developed to understand the natural world and to intervene in it effectively and controllably. But it is not the only form of knowledge. Empirical observation, even without a formal theoretical structure, can identify truths that the scientific method applied later confirms and explains. Tradition, even when it does not understand its own foundations, can preserve effective practices that theory only reaches much later. And the collaboration between those who guard the past and those who investigate the present can open windows that neither of them, working alone, would be able to see. Bald's Leech Book waited for more than 11 centuries. It waited for a moment in which the crisis created by modernity's overconfidence in its own remedies would open space for a question that no one had thought to ask. It waited for a researcher who studied Old English and who wondered if there was something real behind the recipes she was translating. It waited for a microbiologist who, instead of laughing at the idea, decided to test it. And when that moment arrived, after all those centuries of silence on the shelves of a library, what the manuscript had to say was surprisingly relevant, surprisingly current, and surprisingly useful. Bald's medicine will not save the world from the antimicrobial resistance crisis on its own, but it pointed in a direction. It showed that the past may hold answers to the problems of the present. It showed that the arrogance of dismissing what we do not understand can come at a high price. And it showed that the intellectual humility of admitting that we do not know everything, of asking where we had not thought to ask, can lead us to discoveries that our own paradigm could not see. Somewhere in some library, in some monastery converted into a museum, or in some archive digitized by dedicated researchers, there are other manuscripts waiting. There are other recipes, other remedies, other knowledge that generations of human beings accumulated through observation and practice, which were recorded with care by scribes who believed that what they were writing had value, and which arrived at the present day through a path of survival as improbable as that of Bald's Leech Book. If this story teaches us anything, it is that it is worth looking. If this narrative has stirred in you the same curiosity that led two researchers to open a medieval manuscript in search of answers to one of the greatest health crises of our time, then you already know there is much more to explore. Subscribe to the channel and embark on this investigation that spans centuries, cultures, and the frontiers of knowledge, because every episode is a new page turned, a new window opened into what humanity knows and what is yet to be discovered. Let us know in the comments which detail of this story surprised you the most. Share it with someone who believes the future is built only by looking forward, and show them that sometimes the most urgent answers were written long ago, just waiting for the right moment to be read. And like this video, so that more people can discover that the past has more to teach us than we ever imagined.